Welcome to the biology class. In the previous video, we discussed about the cell division mitosis. In this video, we will learn about the stages of meiosis. The term meiosis was coined by Farmer in 1905. Meiosis produces the sex cells or gametes. Meiosis is a type of cell division that occurs in reproductive cells. It is called reduction division because the chromosome number is reduced from diploid 2n to haploid n. Diploid is the condition of two sets of chromosomes. Haploid is a condition of one set of chromosomes. In human beings, the body cells have 46 chromosomes and the reproductive cells have 23 chromosomes. The two sets of chromosome, that is 46, is a diploid one, and the single set 33 is haploid. Meiosis reduces the chromosome number to half, so it is called reduction division. In meiosis, one parental cell divides and produces, this parental cell is diploid cell, 2n cell. It divides and produces 4 daughter cells. The daughter cells are haploid. A single parental cell with two sets of chromosomes produces 4 daughter cells. The daughter cells are haploid. Here the chromosome number is reduced to half. So this division is called as reduction division. Meiosis division consists of two continuous divisions. The first one is heterotypic division or meiosis 1. The second one is homotypic division or meiosis 2. In meiosis 1, the diploid parental cell divides into 2 haploid daughter cells. Here, the chromosome number of daughter cells get changed. So it is a heterotypic division. There is change in the number of chromosomes between the parental cell and the daughter cells. So it is called as heterotypic division. The daughter cells of meiosis 1 enter into meiosis 2. Meiosis 2 is similar to mitosis. The daughter cells enter into meiosis 2. Each one further produces 2 cells. So at the end of meiosis 2, 4 haploid daughter cells are produced. Here there is no change in the number of chromosomes between the parental cells and daughter cells. So it is called as homotypic division. Meiosis 2 is homotypic division. Meiosis 1 produces 2 haploid cells. Meiosis 2 produces 4 haploid cells. So at the end of meiosis, 4 haploid cells are produced. Let us see the meiosis 1 in detail. In meiosis 1, a diploid cell produces 2 haploid cells. The daughter cells are different from the parent cell in the chromosome number. So it is called a heterotypic division. Since there is change in the number of chromosomes of parental cells and daughter cells, it is called as heterotypic division. The meiosis 1 consists of 5 stages. prophase 1, metaphase 1, anaphase 1, telophase 1. After telophase, cytoplasm divides, that is cytokinesis. Let us see the stages one by one. Prophase 1 takes a long duration. In meiosis 1, prophase 1 takes a long duration because it is again divided into 5 stages. Prophase 1 of meiosis is subdivided into 5 stages. They are leptotin, zygotin, packetin, diplotin, and diakinesis. The 5 substages of prophase 1 are leptotin, zygotin, packetin, diplotin, and diakinesis. These substages are not seen in mitosis. This is a nucleus of interphase. In interphase, the nucleoplasm has chromatin threads. The chromatin threads form chromatin network. When the cell begins to divide, the chromatin net becomes coiled. The chromatin coils and form chromosome. The chromatin becomes coiled and appear as long thread-like structures called chromosomes. This is chromatin and this is chromosome. This chromosome is short and thick, but the chromosome of leptotin stage appears as long thread-like structure. This is the cell at leptotin. Here the chromosomes appear as long thread-like structures. Leptos means thin thread. In zygotin stage, pairing of homologous chromosome takes place. Homologous chromosomes are identical pairs. They are similar in shape, size, length. In human beings, a cell has 23 pairs of homologous chromosomes. The first pair are longer and the last pairs are shorter. 2 homologous chromosomes are arranged side by side and begin to pair. In any place of the nucleus, there are 2 homologous pairs. Pairing of homologous chromosomes is called as synapsis. Packetin is the third substage of prophase 1. The chromosomes are visible as long threads. The chromatids are also visible. Each pair now contains 4 chromatids. A chromosome has 2 chromatids, so pair of chromosomes have 4 chromatids. The chromatids are visible in packetin stage. The pair of chromosomes remain attached together at one or more points by X-shaped arrangements known as chiasmata. The chromosomes are attached at certain points. These attachment areas are called as chiasmata. The chiasmata appears X-shaped. The X-shaped attachment areas of the 2 chromosomes are called as chiasmata. The chromatids break at chiasmata and exchange their parts. The broken segments may be interchanged. This is called as crossing over. At the place of chiasmata, the chromosomes break and exchange their segments. This is called as crossing over. Crossing over takes place at the place of chiasmata. Crossing over takes place at packetin stage. So after crossing over, the interchanged parts are seen. Diplotin. The homologous chromosomes begin to separate. Separation starts from the middle and moves towards the end. Chiasmata begin to move from the centromere to the end, resulting in terminalization. The separation of chromosomes starts from the middle and the separation moves towards the end. This is called as terminalization. When the chiasmata moves towards the end, the two chromosomes get separated. It is called as diplotin because the dual nature of the chromosome is clearly seen. After crossing over, the chromosomal segments are exchanged. So the chromosomes are dual in nature. The double nature of chromosomes are clearly seen in diplotin stage. In diakinesis, the nuclear membrane and nucleolus begin to disappear. The spindle fibers appear. Diakinesis is the end of prophase 1. Let us see the quick recap of the substages of prophase 1. In leptotin, the chromosomes appear as thin thread-like structures. Pairing of homologous chromosome takes place in zygotin. Packeting stage involves the exchange of chromosomal segments, that is called as crossing over. The chromosomes begin to separate in diplotin stage. In diakinesis, nuclear membrane and nucleolus disappear. Now the cell is ready to enter into the metaphase 1. The chromosomes move towards the equator. The chromosomes move toward the center. This diagram shows the metaphase cell. Here the chromosomes are arranged at the equator. The centromeres are connected with spindle fibers. The two chromatids of each chromosome do not separate. The chromatids do not separate. The chromatids are connected by centromere. The centromere does not divide, so the chromatids do not separate. In anaphase, the chromosomes move towards opposite poles. In this picture, we see two pairs of chromosomes. In each pair, one moves towards one pole, another moves towards opposite pole. Each homologous chromosome with its two chromatids move towards the opposite pole of the cell. Here the diagram shows the movement of chromosomes towards opposite poles. They move with their chromatids. Chromosomes move with undivided centromere. The centromere does not divide, so the chromosomes move with their chromatids. Telophase is the last stage of meiosis I. The haploid number of chromosomes after reaching their respective poles become uncoiled and elongated. Chromosomes are thick structures. They uncoil and form chromatin net. This diagram shows uncoiling and formation of chromatin reticulum. The nuclear membrane reappears. The nucleolus reappears. After the division of nucleus, the cytoplasm divides into two. As a result of this, two daughter cells are formed at the end of meiosis I. These two daughter cells will enter into meiosis II. Meiosis II is similar to mitosis. In meiosis II, the first stage is prophase II. In prophase, the chromosomes appear. In metaphase, the chromosomes are arranged at the middle of the cell. In anaphase 2, the centromere splits. So the daughter chromatids are separated. In meiosis 1, anaphase 1, there is no splitting of centromere, whereas in meiosis 2, anaphase 2, there is splitting of centromere, so the daughter chromatids are separated. The daughter chromatids move towards opposite poles in anaphase 2. In telophase, the nuclear membrane appears. So, 4 daughter nuclei are formed. After the division of cytoplasm by cytokinesis, 4 daughter cells are formed. So, at the end of meiosis 2, 4 daughter cells are formed. The daughter cells have half set of chromosomes. The chromosome number is reduced to half in daughter cells.